repair. If one small mismatch would get replicated and can cause many problems. Okay, now we got the essential dogma and transcription of DNA. So how does DNA transmit information within the cell from proteins? How do we get from DNA to protein? The essential dogma of molecular biology, which is DNA to RNA to protein. First, what is RNA? RNA is ribonucleic acid, and it is single-stranded but can fold back on itself. It's a ribose sugar, not deoxyribose like DNA. Uracil is in place of thymine. RNA is used to get the message out of the nucleus and into the cytoplasm where proteins are made. The combination of 3 nucleic acids is called a codon. So there are 4 steps to get from DNA to RNA, which is transcription. And the steps are initiation, which is start signal located, elongation, the copying of DNA to RNA, termination, stop signal located, and processing, final changes made. Okay, so first is initiation, where each gene of double-stranded DNA has a coding and a non-coding template strand. The coding strand has the same sequence as the mRNA instructions for protein synthesis. The start of a gene is located by a nucleotide sequence called a promoter sequence, where RNA polymerase can bind. RNA polymerase makes RNA. The promoter sequence has 2 specific regions. One is rich in T and A nucleotides and is referred to as the TATA box. And then there's elongation, which is very similar to DNA replication. RNA polymerase opens the DNA one section at a time. RNA polymerase builds a new strand in the 5' to 3' direction using the template strand, nucleotides added to the 3' OH group. Since RNA is a single-stranded, no Okazaki fragments, many RNA copies of the same gene can be made. Then there's termination, where RNA polymerase will continue along the DNA strand until a terminator sequence is encountered. After this, the RNA polymerase molecule separates from the DNA strand and the new RNA molecule dissociates as well. The RNA is now called mRNA for messenger RNA. Then there's processing, which has 3 steps. It only occurs in eukaryotes. The initial mRNA molecule is called pre-mRNA. The 5' end is capped with a modified G nucleotide. This is called a 5' cap. The 3' has a long string of A nucleotides added to it. This is called a poly tail. These 2 things help protect the mRNA. Then we have part 2 of protein synthesis, which is translation. So the transfer of RNA, tRNA fold into 3 lobe-shaped clover-like. At one lobe resides the anticodon that has 3 nucleotides complementary to the codon mRNA. Across the anticodon is the amino acid attachment site. When amino acid is bound to tRNA, it's called aminoacetyl tRNA. So the first step of translation is initiation, where mRNA reaches the cytoplasm. rRNA binds the 5' end of the mRNA. At a special sequence, an initiator tRNA with an anticodon UAC binds to the ribosomal mRNA complex. This anticodon matches the start codon AUG and carries a AA methionine. Then there's elongation, where the mRNA codon in A site forms a base pair with incoming AA tRNA. Peptide bond is formed, joining new amino acid in A site to growing chain in P site. Ribosome moves at distance of 3 nucleotides along the RNA molecule, the 5' to 3' direction. This is called translocation. Translocation brings the tRNA holding the chain into the P site. A new A site is exposed, and the old tRNA from the P site moves to the E site. This process is repeated many times. Then there's termination, so elongation continues until a stop codon is reached on the mRNA strand in the A site. Previous tRNA carrying the polypeptide chain remains in the P site until a protein called a release factor binds to the A site. The release factor cuts off the completed polypeptide from the tRNA. The polypeptide is now free from the ribosome and will take its shape by folding. Now we're going to talk about mutations and protein synthesis. So causes of mutation. So there are spontaneous mutations, which are results of errors made in DNA replication and occur without chemical change or radiation. There's also induced mutations caused by chemical or physical agent. And chemical agents often have a structure similar to nucleotides and interact chemically with DNA. So example, nitrites or cigarette smoke. And physical mutagens tear through DNA structure. Example, UV or X-ray. Single gene mutation involves the changes in the nucleotide sequence of a gene. It can be substitution or frame shift, both print mutations, or chromosomal mutation, which is a mutation that involves changes in chromosomes and may involve many genes. It can be duplication, inversion, translocation, or large deletions. So we have mutation. One of the mutations is substitution, which is when one nucleotide is replaced with a different one. Frame shift, which is caused by the addition or removal of a number of nucleotides not divisible by 3. This results in a change in reading frame. Translocation, which is a section of one chromosome swap places with a section from a different chromosome. And inversion, which is when an entire piece of chromosome turns around and then reinserts itself. So restriction enzymes were given the name because they restrict the replication of infecting viruses by cutting viral DNA. A specific type of restriction enzyme called a restriction endonuclease is of particular importance to molecular biologists. Restriction endonuclease cut double-stranded DNA with the interior of the DNA strands rather than at the ends. They do so by recognizing the short sequence of nucleotides called the target sequence within the DNA. Next, the enzymes cut the strand at a particular point with the sequence. This point is known as a restriction site. Each of the different endonucleases that have been isolated by researchers recognizes a different target sequence. An example of restriction endonuclease reaction is shown. Recombinant DNA is a DNA that has been prepared in the laboratory by combining fragments of DNA from more than one source. The two cut DNA fragments are incubated with another enzyme, DNA ligase. This enzyme seals the breaks in the DNA, forming covalent bonds between two different fragments. This results in a stable recombinant DNA molecule. Recall that DNA ligase also joins with Zaki fragments when the lagging strand is synthesized during DNA replication. Gel electrophoresis uses an electric field to separate negative charged DNA fragments according to the size as they pass through the gel. The gel is submerged in an aqueous solution called a buffer containing various salts that maintain the pH of the solution. A positively charged anode lies at the end of the buffer bath, and a negatively charged cathode lies at the opposite end. The salts in the buffer carry the electric charge. The gel is made of either polyacrylamide or agarose and provides a porous matrix-like support through that the DNA molecules weave their way through, repelled by the negative cathode and attracted to the positive anode. A recombinant DNA molecule is produced that is composed of the gene to be cloned, an appropriate vector for the host system. A vector is a carrier for the gene to be cloned. For gene cloning in bacteria, the vector is a plasmid. Plasmids are small circular double-stranded DNA molecules found in some prokaryotes. They are not part of the nucleoid region and often carry non-essential genes, although they may carry genes for antibiotic resistance. During gene cloning, a plasmid acts as a vector for the gene to be cloned. To do so, the plasmid must have an origin of replication that allows it to be copied independently of bacterial chromosomal DNA. It must also carry a gene that makes bacteria resistant to a certain type of drug. Most commonly, this gene allows for resistance to the antibiotic ampicillin. Finally, the plasmid must have one or more restriction endonucleosides where the gene to be cloned can be inserted using a standard recombinant DNA techniques. This is necessary because some bacteria will also contain plasmids that do not have this DNA inserted in them. A polymerase chain reaction is an automated method for amplifying specific regions of DNA from extremely small quantities. So the steps in the PCR is that the DNA sample to be amplified is heated to a high temperature, typically about 95 degrees Celsius, so the double-strand DNA is denatured. This is altered structurally by an external stress into single strands. Then the DNA sample is cooled in the presence of two nucleotide primers. The primers are complementary to each 3' end of the DNA fragment to be amplified. The lower the temperature allows the primers to anneal or base pair with the 3' end of the single-stranded DNA to be amplified. The temperature also depends on the nucleotide sequence of the primers, but typically around 55 degrees Celsius. Then, the third step is the DNA sample is heated to 72 degrees Celsius, which is the optimal temperature for Taq polymerase, a certain type of DNA polymerase used for PCR. The isolation of this DNA polymerase from the heat-loving bacteria was one of the key factors that made PCR possible. Like other DNA polymerase enzymes, it synthesizes DNA by the addition of free nucleotides to the ends of the primers via complementary base pairing. However, unlike other DNA polymerases, it can withstand the high temperatures used during the cycling of reactions. The fourth step is that DNA is taken through several cycles of steps 1 to 3, undergoing repeated rounds of denaturation, primer annealing, and DNA synthesis from extraction of primers. Note that the amount of DNA doubles with each replication cycle. Therefore, 1 copy of DNA produces 2 copies after 1 cycle, 4 copies after 2 cycles, and 8 copies after 3 cycles, and so on. Then we have DNA fingerprinting or DNA profiling. So it is identification of people based on their DNA because each of us has unique DNA sequences. It is now called STR profiling or short tandem repeats. Selective PCR primers eliminated for the need for restriction enzymes. So how does it work? Part of the variation between people occur in areas of DNA that do not contain genes/code for proteins. They are highly repetitive DNA because they contain groups of nucleotides that are repeated over and over. They are often called STRs, short tandem repeats. The repeats vary in length depending on how many copies of STR are present. The more there are, the longer the fragment. Primers are designed to amplify specific regions. These regions vary in length between people. The difference in the length is called polymorphism and is unique to the person. Due to the length difference of PCR products, gel electrolysis can be used to analyze and compare samples. There are 5 basic steps: sample collected, DNA extraction, PCR, gel electrolysis, comparison of samples. It is possible that people will share the same banding pattern for small specific areas, so identical twins and close genetic relatives like parent and child or siblings. Applications are forensic sciences, so crime scene investigation, identification of victims, identification of genetic parents. Then we have DNA sequencing, which is the process involved in determining the exact sequence of nucleotides in a piece of DNA, so the nitrogenous basis. It was developed, Frederick Sanger developed the deoxy termination sequencing method in 1975 based on the process of DNA replication that uses deoxynucleotide triphosphates. It is the most popular sequencing method and most easily automated sequencing method. In DNA replication, in order to add the next nucleotide to the daughter strand, the exposed OH found at the 3' carbon of the daughter strand must react with the phosphate group at the 5' end of the next nucleotide. But if there is no OH at the 3' carbon, DNA polymerase 3 cannot add another nucleotide. If a deoxynucleotide got incorporated into the DNA instead of a regular deoxynucleotide, DNA replication would stop because no new nucleotides could be added to the deoxynucleotide. So the Sanger sequencing is single-stranded DNA template using heat, radioactive primers, DNA polymerase, deoxynucleotide triphosphates, and regular deoxynucleotides. So the process is that DNA replication occurs in each test tube. Regular deoxynucleotides are incorporated into newly synthesized DNA, but sometimes a deoxynucleotide will be incorporated into the newly synthesized DNA. The new DNA will stop after the deoxynucleotide because it prevents the attachment of the next nucleotide. Many different length fragments will be created in each test tube. So modern sequencing, the theory is the exact same as Sanger sequencing, but the method is simplified. 4 different colored fluorescent dyes are used to label the 4 different deoxynucleotides in a single tube. All 4 fluorescent deoxynucleotides can be run on one lane on a gel electrophoresis. So the difference between Sanger and modern sequencing is that fragments are still separated by size but show up as colored bands.